Hey there folks, and welcome to the second video for Chapter 28, Nervous Systems. In this video we'll be going over the basic forms and some of the more complicated adaptations of animal nervous systems. Let's get going. Very simple animal nervous systems and things like cnidarians and echinoderms, so imagine animals like jellyfish and starfish, have a simplified nervous system which we call a nerve net. It's called a nerve net and not a formal nervous system because it's not really centralized in one area of the body. These are animals that don't have identifiable heads. There's no brain. And so there isn't really a nervous center for the nervous system. So in this case, we have a nerve net that spreads out throughout the animal's body. In this case, down a hydra and down into its tentacles, or around the center ring of a sea star and down its legs as well. Starting in slightly more complicated animals like flatworms, we have something called cephalization. Cephalization is the formation of an identifiable head. And at this point, this is where we start to get species that have what we would define to be a brain. You've got central ganglia. It's not quite the regular animal organ that you might see in dissection, but it is a concentration of sensory organs at one end of the body, and likewise a concentration of nerves, of neurons, at one end of the body. The more specialized and more cephalized animal evolution became, the more centralized the central nervous system became. So from a very simple diffuse nerve net and say a hydra, which is a cousin of a jellyfish, until we get to cephalization, where we first get a head in flatworms, we then achieve something that's much more specialized in things like insects, really complex invertebrates like insects and octopi, but also vertebrates like birds, mammals, and reptiles. In these species, we see centralization, where we don't just have a head, but the nervous system is centralized there in the head. This is where we start to call it a central nervous system. It is centralized in the head or at the head end of an animal. Likewise, at this point is when we see a nerve cord that starts to connect nerves in the centralized central nervous system to the rest of the body. And we first start seeing that here with the two nerve cords in a flatworm. And again, as animal evolution became more cephalized and more centralized, these two nerve cords eventually became in vertebrates the spinal cord. In vertebrates, we don't just have the spinal cord, we have a lot of adaptations to help protect this centralized nervous system. The spinal cord is there to help connect the brain down into the nervous system and the rest of the body. Basically to connect the center of the centralized nervous system down into the peripheral system that's helping to run the rest of the body. In vertebrates, we call that centralized nervous system the brain. That center of it where all of the thinking and learning and decision making about how to handle a stimulus, where all of that goes on. And because this is such an important but sensitive part of our system, we have a lot of very specific adaptations to protect it. In fact, most of the rest of these terms are all about how we protect our brains, how our bodies and our nervous systems are structured to protect it. For instance, our blood does not make any direct contact with our brain. This is an adaptation to help try and prevent large molecules which might be toxic from ever contacting the brain. So things like waste products from the rest of the body that may be carried by the brain on its way through the body, they will never make contact with brain tissue. In a normal capillary, where the capillaries are doing the rest of their job elsewhere in the body, blood and substances in the blood can diffuse out of the capillary through pores right into the surrounding tissues. No problem. This isn't possible in a brain capillary. There's a whole extra layer of protection made by cells called astrocytes that helps to prevent regular diffusion in and out of the capillaries and into the brain, because that might actually put the brain in contact with potential waste products or toxins that the blood might be carrying. Right, this layer of protection is called the blood-brain barrier. The way we do get things like oxygen and nutrients into our brain is through the cerebrospinal fluid. As the substances from blood passes through the astrocytes, either directly through lipid-soluble substances, or if it's actively transported through the cell, it's filtered. This filtered fluid that makes it to the other side of the astrocytes isn't blood anymore. Too much has been left behind for us to call it the same thing, and it gains a new name. This is cerebrospinal fluid, and it is the liquid that bathes the tissues of our brain. It also has a direct connection to the central canal inside our spinal cord. So say if you ever watched ER or another House or another hospital drama on TV, if anyone ever has to have spinal fluid drawn out for some kind of horrible, painful test, this is the fluid they're going for. But they're not going to try and take it from the brain. That would be a horrible idea. They're taking it from the spine. The cerebrospinal fluid is found both in the cerebrum, in our brain, and in the center of our spinal cord. That's how it gets its name. The last major adaptation for protecting the brain that I'm going to cover is the meninges, and it's not just present in the brain. It actually wraps around as a protective layer around our brain and our spinal cord, around the entire central nervous system. And it's just a protective, cushioning extra layer of tissue. You can find it around the brain, between the brain and the skull, to help cushion the skull, and also around the spinal cord, between the spinal cord and the vertebra, to help cushion the spinal cord. Inside the brain and in the spinal cord, we find a mix of gray matter and white matter. They're called this because they have a generally lighter or more gray cast to them if you ever saw them exposed to daylight. White matter is primarily made of the axons of neurons. It's whitish because the myelin sheath that wraps around axons to help speed the reactions has a whitish cast at that distance. Gray matter consists mainly of nerve cell bodies and neuron dendrites. That's all that is. Inside the central nervous system, there are two major forms of the system: the motor system and the autonomic nervous system. The motor system of our central nervous system is chiefly concerned with connecting the central nervous system to our skeletal muscle. All right. Anytime you can have your brain send a signal to your foot to kick your foot or twiddle your toes, you're using your central nervous system, because you're accessing skeletal muscles that our brain can access and exert conscious control over. The autonomic nervous system connects our brain to the other two types of muscle, to smooth and to cardiac muscle. So, for instance, to say, control heart beating, the heart rate, or the rate of digestion, how muscle fluctuations and movements can help move food through our digestive system, that sort of thing. The autonomic nervous system is broken down into three different categories: the parasympathetic, sympathetic, and enteric systems. The parasympathetic nervous system is basically the rest and digest setting for our nervous system. It primes the body for activities that are all about either gaining or conserving energy for the body. So stimulating our digestive system to break down food and take up nutrients, preparing the body to eat, preparing the body to rest, or getting the body to rest. The sympathetic nervous system, on the other hand, is fight or flight, exciting the body, amping up the heart rate, damping down the digestive system because we need to maybe redirect all of our energy towards movement, towards running or something else. Stimulating our adrenal glands to make things like epinephrine and adrenaline to get the body moving. These are extremes. Sitting and doing nothing but eating and running for your life are rather extreme activities, and you will almost never be completely managed by one or the other, but by both the sympathetic and the parasympathetic, bouncing back and forth, activating a little over here and a little over there, and downplaying a little over here and a little over there. Between the two of them, we tend to achieve some form of balance that dictates what sort of state of excitement or relaxation the body is in. At the far other end, the enteric division, the last subdivision of the autonomic nervous system, primarily manages neurons in the digestive tract, the pancreas, and the gallbladder. It also helps the activity of smooth muscles that produce peristalsis. That is the movements, those fluctuations in muscle movement I was talking about earlier that can help ease food through our digestive system and gradually push it through our intestines so that we actually get an opportunity to digest and to absorb food. The last section I'm going to cover for this chapter starts getting us into the next section. This is the human brain. And not actually just the human brain. This is a mammalian brain. We'll get into the human brain in the next video. But this is how all mammalian brains are structured. Actually, even more general than that. This is how all vertebrate brains are structured. So this is the general structure of not just mammals, which we know and love well, but fish, bony fish, reptiles, birds, amphibians. We all share these basic brain anatomical characteristics. Our brains are made up of a forebrain, a midbrain, and a hindbrain, and each one of these regions does a particular job for the central nervous system. For instance, the outermost layer of the forebrain, the cerebral cortex over here, this is this outermost layer where you actually can see like the wrinkles in the topography of the brain. The more texture there is to the cerebral cortex, the more layers there are. Generally speaking, the more developed the brain is for perception, for voluntary movement, and for learning. We tend, because this is the job of the cerebral cortex, we tend to see a much more developed, enlarged cerebral cortex in mammals, which are more likely to use things like learning, than in other vertebrates like fish or reptiles. And even amongst mammals, mammals that do more active learning tend to have a much more enlarged, textured, complex-looking cerebral cortex than, say, rodents, which don't do that much active learning. Alright, folks, I'm going to leave the vertebrate brain there for right now because the last section of this chapter is going to have us concentrating on it in greater detail. But I'll see you then. Bye, folks.